היי. לא, זה בסדר. לא, זה טוב שהגעת זה, אחרת לא היה בכלל שום קורס. כן. Okay, can someone close? Hold on. Hi, so how are you? How was Christmas vacation? There was a Christmas vacation. That was why last time we didn't have class. So I hope you played with a snowman and all the things you do in Christmas. And yeah, so last time, this is not actually last time, so we will go over the paper by Eric and L, which is Eric and L is one of the, I would say, like the pioneers of neuroscience in general, in molecular neuroscience in particular, and that he studied really in a very meticulous manner. If you follow his career, he started with a very simple model organism. He studied the aplysia, and just went step by step each time in his research in a very gradual and very consistent way. But in the end, he discovered a lot of very important like mechanisms of molecular, like what underlines the molecular basis of memory, and specifically long-term memory. So I gave you guiding questions for this paper. So first, what is Krebs? Can someone tell me? Let's start from Cree. What is Cree? I gave you a hint, which is a DNA sequence. So what is it? It's a gene? No, Cree is not a gene. It's more, so it's a short for cyclic AMP response element, which is, it is a DNA sequence, but it's not a gene. It's a regulatory element that cyclic AMP responsive element binding protein, which is Krebs, knows how to bind to, and it can induce the transcription of genes. Okay, so it's like sitting in the promoter regions of certain genes, you have a cyclic AMP responsive element, and this is actually a thing that we see more and more today. A lot of time, if you have this sequence sitting in the promoter of your gene, it's very probable and there's a high probability that this gene will be regulated by neuronal activity in general. So this is one of the things that Kendall gave us from that. And I will not define E-LTP, L-LTP, and E-LTP, because you could just read about it. And in what way is the RB subunit different from the wild type unit of PKA? Yeah, so it's what we call a dominant negative form, meaning that if it's present, even one allele, one copy of it, even if it's present in the cell, it will cause a reduction in the activity of the PKA. So they used it in order to diminish the levels of PKA, but not to delete it completely. And so the rest of the things that we'll get to in the. So the research question that they ask here is, first of all, what is the relationship between memory and L-LTP? Because L-LTP is a physiological phenomenon, right? So you see, it's a long-term, very long-term potentiation. So it's like it's something that you see in a slice or in our apparatus, and today people are struggling to show it in vivo. I don't think that they managed to until now, but it's something that you see in the serial responses, and it doesn't have to be related to memory in any way, but it looks like it feels like it should be related to memory, but you need to see in which way, and also to define the role of pKa, which is of this enzyme that phosphorylates, that has the power to phosphorylate. Again, that's why it's a kinase. It's a protein kinase A. And what is its role for long-term memory? So when I look at after I read a paper and stuff like that, a lot of time I make like a mental flow, or I see how the research has progressed along the paper. So, and this is like the study summary that I did for this paper, is that what they did is, the manipulation they did is to reduce the activity of pKa only in the hippocampus, or at least they tried to do it only in the hippocampus. Then they saw that L-LTP was decreased in the CA1 region of the hippocampus. Then they went to the behavioral test and saw that animal exhibited long short-term memory but deficient long-term memory, which is the connection between L-LTP and long-term memory. And it correlated, or it showed it was similar to what happens to animals that are treated with protein synthesis inhibitor. And from that they deduced the connection between the two, but obviously there is no causal relationship in this step. So what actually did they do? They used transgenic mice. In principle, in this paper, they show results from 2 transgenic mice. Like I told you, they show what they did is that they overexpressed the dominant negative form of one of the subunits of the protein kinase A, and this is how they did the construct. So the reason that I'm showing you, like, this is one of the first demonstrations of a genetic construct, and nowadays these constructs look very different. For example, do you remember what's the length of this promoter region of this construct? So it's 8.5 kilobase. It's a very the actual sequence of the DNA is very short, but the promoter region, like what they did in the past. So we return to the scheme of what is a gene. So a promoter region is this entire, like, a region that is not very, you can say, not very well defined in terms of length, that comes before or upstream of the gene itself, or it's part of the gene, but of the part of the gene that will be transcribed into mRNA. And back then, it was very hard to define which sequences of the promoter are important. So the promoter region is important for the RNA polymerase to attach to and start transcribing the gene. So the transcription of the gene starts from this location, the 5' UTR, up to this region, but the binding of the RNA polymerase occurs here. And back then, it was very hard to define which part of the promoter is important for transcription. So they had to take like huge chunks of DNA that they knew, like, that are behind the CAMK2A gene, and just put them to make sure that they have all the elements that are important for the transcription of that gene. What did they take a CAMK2A promoter? So back then they said that it's specific to the frontal and hippocampal regions. Like, it's a little bit loose now we have, but again, this is one of it's 1997, so we'll give them slack. But the thing is that it's one of the first uses of an exact, like, what we call a marker gene. Okay, so if you have a gene that you know that has a specific pattern of expression, for example, what they show here is is a special type of staining that's called in situ hybridization. So they're actually showing the mRNA or the mRNA that comes from this. But in general, if you have a gene that you know the expression pattern of it, for example, if the gene is expressed only in the hippocampus, only in the frontal regions, only in the cerebellum, for example, then what people used to do in the past is that you would say, okay, I will take the promoter region of that gene, and I'll put my own gene in front of that promoter region, and in that way it will cause a specific expression of my gene that I'm interested in these regions. Because the idea was that the promoter regions are like the ones that determine the specificity. So I'm not sure if you're aware, but for example, we have about 20,000 genes. We'll talk about it actually in next chapter, like the chapter after the G protein coupled receptor, that there's like 20,000 genes in our genome, but actually each cell, if you're talking about neurons, they can express something like between 7,000 and 11,000 genes. They don't express all the genes. And even if you go down to the level of the single cell, it's even less. So the fact that cells in the hippocampus, for example, will express a certain gene and not another gene, and this is what causes this specificity. And actually nowadays there are like a little bit cleaner ways to do this manipulation, but it's more or less the same principle. So they use the CAMK2A promoter, then they put an intron here. Do you know why they put an intron here? I can tell you that this is just a fragment of this is a fragment that's not needed. This is going to be cleaved out. Okay. So, but why do that? You know?
They said it was to enhance expression of transgenes. Like that's what they put in there.
Yeah. So again, nowadays we have better ways to enhance expression, but theoretically what they did is that they introduced an artificial construct into a cell. And a lot of times there's this notion that the cell is something that is it's highly regulated. The cell knows in essence, like the cell has a lot of ways to know if the transcript that was inserted into it is something that it needs to express or needs to degrade or something like that. So you need to add a lot of elements that will add stability and also tell the cell that this construct is okay and you can now treat it as your own genes. And one of these things is what they did in the past is adding introns. And this approach came from the fact that people believed, and also they saw it in experiment, that if a gene is processed, meaning that the introns are getting cleaved out, then it enters like a pathway in the cell that tells it if it's processed, then it means it's okay. Like if it undergoes processing, then it means that this transcript is okay and can be expressed and in the end get to the level of the protein. For example, there are elements that are left on the mRNA between this step that the introns exist and after they're cleaved out. So there are special elements that remain in these junctions that probably help, for example, for shipment outside the nucleus and add again to the stability of that construct. And then they have the actual construct, what you call cDNA of the actual gene that they wanted to express, which is the subunit of the pKa, and then they added a poly A, like poly A. It's more like a poly A signal. So again, this is one of the elements that you want to add to your gene in order for the cell to add a poly A tail to it, and again contribute to its stability. But we're talking about that in the last chapter more. So here is actually the proof that you have the expression, and you can see that the expression is not really totally specific, but you have a very strong expression of their construct in the hippocampal region, which is here. Okay? And in the wild type you have nothing, obviously, because you don't have this construct. So they wanted to show that they can detect their construct and not get like artifacts, and that the construct is expressed in a strong way in the hippocampus. Okay, so after they showed us their experimental model, now they started testing it to show what is the effect. So the way that they did it is again a method that people don't use anymore, but it's a way to measure directly the activity of pKa, or the activity of an enzyme. What you actually do is that you extract like a sample, or you take a protein sample from the brain of these mice, and you put them together with this special plate that has a substrate, meaning that something that the kinase can phosphorylate on this special plate that you can then detect a signal from. In this case, it's a radioactive signal. So nowadays people do it with fluorescent methods. The radioactive methods are less preferred today, but they're actually very good and very efficient. They're just not used because they're not very safe. You don't want to start messing with radioactive things. But up until recently, it was still used. So the idea is that if the kinase is more active, then you're going to get more phosphorylation on the substrate. And the way that you detect this phosphorylation is that you probably remember that kinases, they take their phosphate from ATP, from a molecule of ATP. And what they did is that they incorporated into the solution that they do this reaction, ATP that has a radioactive tagged third phosphate. So what the kinase actually does is that it transfers the radioactive phosphate from the radioactive ATP to the substrate, and in the end what you get is that you can measure in each one of these wells the level of the radioactive radiation, and from that get the activity of the enzyme. So what they measured here relative to the wild type, and they show that they really have a reduction in the level of the basal level of the pKa. And when they added cyclic AMP, which is an activator molecule, so we'll learn about it in next class, but cyclic AMP is what we call it's a type of secondary messenger in signal transduction process in the cell, and when it attaches to pKa, it causes it to be like hyperactive. It causes an increase in activity of the pKa. So when they add cyclic AMP, all the pKa's become much more active, but again there is a diminishing like effect of this dominant negative form that they expressed together with a regular form of RAB of pKa. So here you see the dominant negative form that causes an inhibition or a decrease in the activity. First of all, I wanted to ask you, do you think that this effect is very dramatic? This effect? What? Yeah, maybe it was very consistent. I don't know, maybe it's very significant, statistically, or if they will increase the groups, maybe it will be more. But do you think it will be stronger? If you increase the groups, you would get an effect that is here. So in the end, you can claim that the diminishing activity of although in the basal level, which is interesting because in the basal level they might have affected the activity in 50%. Here, when the enzyme is actually activated, then they got a reduction of 10%, or maybe 20% in the activity of the enzyme, which is not very dramatic. But again, it's interesting because it tells us that first of all there might be an effect on the basal level also, not only with the cyclic AMP, and also that you don't need a lot in the molecular level to cause an effect that will maybe be dramatic in terms of memory and the phenotype that they saw. So what exactly did they do? This is their experimental setup. Back in that time, it was very, very classic experimental setup that because this whole loop of the hippocampus is very well known, the connection between all the different types of cells is very well known, and you can put like an electrode that sits on the what it's called the Schaffer collateral, and stimulate here, and then record from cells that are sitting in this layer that's called CA1 of the hippocampus. So it's a very classical paired pulse experiment, but in this case, what they did is that they applied what they call tetanus stimulations. So it's like repeated stimulations that are very fast. That's why it's tetanus. And interestingly, when you apply one train of tetanus, then you get a potentiation, and a potentiation is an increase in the response of like this cell, or the increase of the response of the synapses here, when you stimulate after like the pulse. So you see that the difference between this and this is exactly the long-term potentiation. It's long-term because you see that scale is very long. It's like minutes in terms of neuroscience. It's very long. But still, when you talk about late long-term potentiation, it's interesting that there's a difference between one train and 4 trains. Actually, in this figure, it looks like a very continuous thing, but actually what they show here is that it's not. They claim that there is a fundamental difference between these two processes. If you put 4 tetanus stimulations or 1 tetanus stimulation, which is interesting. So, what they wanted to show us next is that, okay, they showed us their animal model, and they showed us that the activity of PKA is reduced, but then they want to show that short-term plasticity or early long-term plasticity is unaffected. And they show us, I think the more convincing result that they show here is that the differences between the transgenic mice and the wild type is non-existent if they give like 1 train of tetanus pulse and 2 trains of tetanus pulses. So you see that there's almost no difference in the size of the response. And also they individually measured like the response of the fiber volley amplitude, like the stimulation that they gave on the Schaffer collateral, and the response in the postsynaptic cell. But actually, one of the things I didn't like about this graph is that I just saw it today, is that when you actually look at it, it does look that there's some kind of difference between the wild type, right? So, like nowadays, this will not go without showing any correlation or stuff like that. Like they did some kind of weird statistical test that I'm not sure is valid, but so I didn't have actually time to test if it's really significantly different or not. But I reconstructed the results, and I think this is like more informative in this. And here you can see like you actually don't see never mind, but these 2 lines are from the transgenic, and this is the wild type. So I think that there might be a difference that is also probably statistically significant, maybe, at least between the green and the black one, but still there is a difference in the slopes between them, which they didn't report, which is a little bit disappointing. But anyway, what is amazing about these results is that you would say, okay, you gave 1 train of tetanus pulse, then 2 trains, then 3 and 4, whatever. What does it matter? Like you just get a higher response. But actually, what they saw is that when they give 4 trains of tetanus pulse, they really get a fundamental difference even in the early response to the. So it's not like something that is additive. It's something that is fundamentally different between the wild type and the experiment animals and the transgenic animals in the way that they respond to 4 trains of tetanus pulses, and in their ability to maintain this form of plasticity. So right in the beginning, they don't have actually the capability to get to these heights of potentiation, like the wild type. But also later they lose this potentiation, whereas the wild type maintain it for, they say, can maintain it up to 24 hours after even, or 8 hours after, because they're only doing it in slice, so it doesn't survive that long. And here they show the responses, like the individual responses. I think it's 180 or 120. I don't remember. Like that. This is before the tetanus pulses, and this is after tetanus pulses of the wild type relative to the RAB, RAB1, and RAB2. So you see, and what they actually measure here, the FEPSP slope, is actually this slope here. Okay, so you can see that that the slope is much sharper in the wild type relative to the RAB1 and RAB2. Do you see something else here that bothers you, or you're okay? What about the responses before? Okay, let's look only on these lines. Are they the same? No, they're actually not the same, which they ignored, but there's no it's not a very bad thing, you can't acknowledge everything, but it's something that I think got in their way, but they just chose to not talk about. But you can actually see that the baseline activation of the RAB2, for example, is much stronger. Like the effect of the synapses, the activation of the Schaffer collateral on the postsynaptic sinuses in the RAB is actually stronger in the baseline level than in the wild type. It actually looks like it's already potentiated in a way, and also the RAB1 is a little bit slightly higher, or gives a higher response relative to the wild type, and it just comes to show you that you need to choose how to show your results. Okay, so you need to choose what to show, because if you really measure the FEPSP slope, then and that you don't measure the peak, for example, then it's very different. Like the change in the slope from here to here is nothing, and the change in the slope from here to here is very dramatic. So I'm just saying it as a point of thought for your future papers. But so to close the deal, also today it's like that, and not only in the 97. So you have to show, if you want to show a very significant result, it's better to show it to go all the way to the behavioral studies. Okay, so again, they started with the molecular. They showed the molecular logic, then they showed the electrophysiological logic, and now they want to show us that there is a difference in behavioral tests. So the Morris-Water maze. Do you know the Morris-Water maze? You should have read about it anyway. So it's a very common test to test spatial memory for mice and learning in general, and you can see that there's a large deficiency in the target quadrant, which is the one that the mice should be in, between the wild type that choose this target quadrant relative to the transgenic animals. And they also did another experiment, which is contextual memory, which they again claim that contextual memory is a task that you need your hippocampus for. And in this case, when they gave the animals foot shock in the context, in one context, and then they tested, and then they for example, a specific room or specific cage, and then 24 hours later they put the mice there. They tested how much they reacted as if they're about to get a foot shock, and they saw that actually what happened here is that the transgenic animal completely forgot what was in that room, okay, relative to the wild type that behaved as if it was right after the foot shock that it got last time. So this is comes to show that their long-term memory is somehow so here it's hard to get the kinetics, and here they said that the long-term memory is affected, but the short-term memory, that how they reacted when you put them in a room 1 hour after you gave them a foot shock was intact. So it's a point for the long-term memory. So, and then what they did is that they wanted to show if the effect is something specific to the hippocampus, or it's something like in general, because you can always claim, and I always say in biology, it's very easy to ruin something. Okay, it's very easy to screw up something. Like biology is something that evolved through millions of years of evolution, and if you tamper with something, you will probably ruin it. Okay, like the best papers show some kind of improvement, like in some kind of feature, and there's a lot of papers, 99% of all papers show disruption of something. But it's very easy to ruin something, so they wanted to show that what they didn't just ruin the general ability of the, or all sorts of types of memory of the animal, so they chose a task that is regarded as a amygdala task and not as a hippocampal task, which is conditioning actually. And they show that in this task, it doesn't matter if it's 1 hour after the conditioned stimulus or 24 hours after the conditioned stimulus. There's no difference between the wild type. So this ability is kept because they claim that it's an amygdalar property. And then they also wanted to show that the size effect that they saw is similar to what you get if you stop protein synthesis. So they added a substance that is it's like a type of antibiotic, but it's a type of substance that you can give the mice that temporarily stops all protein translation. So it's like it's affecting the ribosome, and it blocks the ability of the ribosomes to transcribe new protein, to translate new proteins. And what they show here is that when you stop the ability to translate new proteins, you actually get exactly like a very similar effect to what they got here. So this is it. This is wild type mice with the drug, and wild type mice with just saline, like normal saline. And they see an effect in the size that is similar to what they got in the transgenic animals. But in this time, it's in both the contextual memory and the conditional memory, because they did they claim that they did like a total, you can say like a total effect that affected also the hippocampus and the amygdala in this case. Okay, so I think it's one of the more classical papers that really added a lot to the way that we understand memory, and also associate it to protein translation in general. Obviously, there's a lot of things that we know now today that they didn't know in the past regarding for the kinetics. And one of the examples for the things that they didn't know in the past is the model that they offer in the end. So first of all, so their model is pretty, here it looks very elaborate, but in essence, what they say is that you can either, if you activate a synapse and you have a lot of calcium entering, then this can activate a signal cascade that will cause an increase in cyclic AMP, and then PKA will be activated, move to the nucleus, activate CREB, which will bind to CRE, and cause transcription and then translation of new proteins that can induce long-term potentiation. Okay, so it can either come from a neuromodulatory response, like for example, from like dopamine or acetylcholine or something like that, or it can come from a very strong activation of a synapse that will cause NMDA channels to open, which you already know, and cause an influx of calcium that can also affect the production of cyclic AMP and the activation of PKA. But in this model, is there something? Do you think what's bothering you about this model? Is this model bothering you?
Too complicated.
Complicated?
In a way that, even when we try to explain something, it should be a simple form. And they have many requirements.
Okay. But I'll ask you like this: like, does a neuron look like this?
No.
Okay.
Let's see a real picture.
No, you already saw real pictures of neurons. I'm not going to show you a real picture of neuron, but a neuron doesn't look like that, especially this nice line over here. Okay, like the signal that has to come, you know, especially like from here, then to here, then to here, and then modulate, or actually it has to come back to this synapse and change something here, is a little like they made the neuron like short and stubby for a good reason, so they won't have to put their lines like in a way that doesn't make any sense. But back then, so they didn't know a lot about what I told you, like for example about local translation, that proteins can be made locally at the synapse, and also it doesn't fit because it has to be through the nucleus. Okay, like so it really bothered I think Kandel back then, the fact that you have to like go through the nucleus, and nowadays we actually know, like even Kandel in a more recent, not very recent, but from 2001 review already started extending his synapse a little bit, but still, obviously one that developed his entire career on the effect of Cree and Krebs and all this thing will not show you a real neuron because it has problems with that model. But even in a more recent review from 2001, he already started giving more elements that are sitting locally in the synapse. So you see the synapse here is like empty. There is nothing here. The synapse doesn't do anything. It just gets responses, and even the calcium here is directly going outside the synapse and then affecting. Nowadays, we are very much aware of the fact that probably everything is in the synapse. Okay, we're not saying that the Cree element and Cree element binding protein are not important. They're very important, but for like the general activity of that neuron, more. And probably the effect that he saw back then is a local effect of the activation of PKA, and that protein translation that he saw that the deactivated was actually the activation of local protein translation. Maybe, I don't know, because nobody did like the differentiation between the two. And but nowadays we feel, and we have also evidence that all the elements that you need to make new proteins and to respond can happen locally in the synapse. So it doesn't have to go through the nucleus. Okay? There are probably pathways that go simultaneously. Like what happens when a synapse gets really activated, there are two pathways. There is a pathway of local translation, and also there are signals that come to the nucleus. And probably new mRNA, new proteins can also get shipped, but this is a much can get shipped to the synapse, but this is a much slower process. Okay, so one other thing that I wanted to show you is that I think that the reason I like old papers in general, and also for your assignment, people ask me if they can choose old papers. I say it's better to choose old papers because I think that they're written in much more clean language. I don't know if you're reading a lot of articles, but I think they come in much cleaner language than what you can find today. And to explain that, again, one of the things that I'm trying to show you is like how to maybe take from that to how to write your own abstract for your own paper. And one of the nice things that I like to do is also like to ask questions, specifically like you're having a conversation. So I just took their abstract and divided it into answering questions. So first of all, why? Like why do what you're doing? Is it to explore the role of protein kinase A in late phase of long-term potentiation and memory. Okay, how are you going to do that? We generate transgenic mice that express RAB, an inhibitory form of the regulatory subunit of PKA, only in the hippocampus and other forebrain region by using a promoter of gene coding calmodulin protein kinase 2. And what did you find? In these RAB transgenic mice, hippocampal PKA activity was reduced, and L-LTP was significantly increased in CA1 without affecting basal cell transmission or the early phase of LTP. So that's interesting. What else? So moreover, the LLTP deficits were paralleled by behavioral deficits in spatial memory, but not short-term memory for contextual fear conditioning. And now the most important question that you need to ask yourself in science is, so what? So why, like, and I think that every one of us, and I also try to ask myself, sometimes the answer that I get for myself is not good enough, but I think you need to, as a scientist, you need to ask yourself this question continuously. So what? These deficits in long-term memory were similar to those produced by protein synthesis. Thus, pKa plays a critical role in consolidation of long-term memory. This is like the bottom line of that research. Okay, so we'll take a break, and we'll return 5 minutes before 4:12 to continue. Hi. Yeah?
So I was very responsible and I was expecting an email and I just completely forgot to sign it up and do tomorrow. So instead of emailing you, can I just ask your little computer
What?
for this paper?
It's not due for which?
It says one.
No.
Oh my gosh. I'm American.
So?
1-2 is January 2nd.
Ah.
So I looked just now and I just had a panic attack.
No.
Okay, fine. So I can email you. I don't need to bother you.
No. There, you have 2 months.
2 months.
Okay.
No, 1 month.
You have
Like
1 month.
today.
Yeah, you have 1 month.
Okay,
Yeah.
so then I don't need to
1
bother
month.
you. I can email you. I was gonna do it for tomorrow, but I
Oh.
figured instead of waiting for you to go back and check your email.
No.
They're not approved.
No.
Okay.
No, I would have told you. I would not do that.
Wait, so you gave an email of everything. I said, okay, we're gonna go over to class, and maybe I'll get an email. Fine. So I put it out of my head, and I just went to go just
No.
open the article again.
I just presented it 4 days ago.
Great, and I wasn't able to come to that class.
No,
so
but I will not give you an assignment for 4 days.
No, I assumed I just missed it. And I missed an email somewhere, and it was due tomorrow, and I was gonna do it for tomorrow.
But you're right. I should change the.
No, it's me. It's
No.
I don't know if it's anybody else also, but.
I don't think that they were worried.
Right.
They don't look worried. Probably didn't read the presentation also. No.
Okay. Thank you. I'll email you.
Okay.
Yeah, he did a whole project.
No, you have 2 months.
February.
No, February 1st. Ah, yeah, you have 1 month. 1 month.
Either way, I have more than 1 day.
Yeah, you have 1 month. Okay, so this chapter that we just started last class is on G protein coupled receptors, and it's first of all, I think it's a critical chapter that you will get from that I think it's important to get from this course, because it describes like secondary or more long-term responses that cells can have in response to like communication between the cells. And from that we started like this chapter with an overview of signaling molecules and cell surface receptors. But in general, in more simple terms, signaling molecules and or in general, like what is cellular transmission or cellular signaling is just, in other words, saying cellular communication. Okay, it's a way that cells talk to one another, and the way that the messages are interpreted inside the cell itself. So this is just to show that there's a lot of different types of pathways that a signal can be interpreted, and there are also many types of signals that that cells can communicate between one another. So, and there's enormous like a variety of chemicals that can be involved in this communication, like amino acids, lipids, basically all the types of molecules that you know about can be involved in cellular communication or communication between cells. And in the level of the ligand, or how we define these the molecules that the cells communicate through which, we said that there's 3 different types of categories that we can divide these signaling molecules. Molecules that are what we call endocrine, that are signaling molecules that act on target cells that are different from their origin or the origin of their synthesis. For example, hormones. These can actually come from outside the body itself. They don't have to come from the body, so they have like a long-term or large effect. Paracrine signaling, which is close proximity signaling, and neurotransmission is a type of paracrine signaling when 2 cells are in close proximity and talking to one another. And autocrine signaling is a subclass of signaling that the cell is actually talking to itself, or signals that the cell or types of molecules that the cells release and recept the same signals. So, and some of the signaling molecules can act both at short range and long range. For example, epinephrine can be a neurotransmitter and a hormone. Okay? So, and one of the examples that I wanted to give you that is like a general example of what is signaling before we dive into the G protein coupled receptors is that, for example, during development, these signaling molecules can, like with a very simple model, we can start to explain how biological boundaries are formed. And I showed you this simple figure from a review from 2005 in Nature, which shows that, for example, if you start, because every organism starts from a position where all the cells are equal to one another, and then randomly one of the cells can like choose to start expressing like a signaling molecule. In this case, it could be like a hormone or something. And then, like a cell, just a second. So, a cell here can start expressing this molecule, and then what will happen is, because of the distance, you will have a decrease in the concentration gradient, and then cells over here can react differently to cells over here regarding to in the proportion of how much molecule they saw around them. And this can lead to secondary effects that will lead to ongoing effects that in the end will cause differentiation of the cells and starting to development of clear boundaries between the cells. What will be like, for example, in the nervous system, we know of a very important molecule that's called sonic hedgehog, in which where there's a high concentration of sonic hedgehog, you have the neural crest and the beginning of the neural tube that will differentiate into different areas of the brain. So it's a very important molecule and determinant to which region of the brain will be where. And I showed you that sonic hedgehog was discovered in fruit flies, and this is how the mutant looks like, and this is what it got the name. So it has very severe developmental issues, and it has this porcupine structure, so this is why you got the name sonic hedgehog. So in intercellular signaling in general, we find that there's a very, very high specificity between the receptor and the molecule that it has to recept, or the ligand itself. So we're still in the level of the ligand. And so, but in contrast, there are a lot of different types of receptors that can be sensitive to the same molecule. For example, acetylcholine, if we're talking about muscles, so it causes, the receptor here will cause muscle contraction. In the heart, it will cause slowing of the heart rate. And in the digestive system, it will cause digestive enzyme to be secreted in the pancreas. Okay, so it's all the same molecule, but it will have very different effects in different areas of the body because of the different receptors that get it, but the receptors themselves are very highly specific. So it's not that a receptor for acetylcholine will not recept like dopamine or something like that. Okay, so there's a very high degree of specificity. So moving on to the level of the receptors. So although, and also in contrast to the fact that there is a lot of different cellular, or a lot of different receptors that can be in different organs of the body, in the end, what happens after the activation of the ligand, and after it's been absorbed or detected by the receptor, the cellular response will a lot of times be very similar. Okay, so for example, what I showed you here, that slowing down of the heart rate and then secretion of digestive enzyme in the pancreas, can, when you look inside the cell, often it will the response will be mediated by the same molecule. And here I show the example of cyclic AMP, which you saw in the article of Kandel. So it's not a special thing for the pathway of the the pathway that we saw in synaptic plasticity, like in the article of Kandel. This is actually a very common feature, a very typical signal transaction molecule. And this type of molecule is what we call second messenger. So the first messenger was the ligand, the molecule that before, or for example, acetylcholine, the example that I gave you earlier. And the second messenger is the molecule that does the work inside the cell, or relays the signal inside the cell. And one of the most common ones is cyclic AMP. It's a derivative of ATP, actually. It just has like this phosphate that is linked to the sugar backbone, and this is why it's cyclic. But you have also a lot of different types of molecules. For example, calcium, like you saw again in the Kandel paper. Calcium can be a signaling molecule, and it can be a secondary messenger by itself. For example, in muscle, it can cause contraction. In endocrine cells, exocytosis, and in nerve terminals, like we actually saw, it causes release or exocytosis of a neurotransmitter from the synaptic terminal. So, and what actually the response to cyclic AMP does is that it activates enzymes, often kinases. So again, it's very similar to what we saw in the Kandel paper, and that the activity of kinases, like in the Kandel paper, phosphokinase, protein kinase A, is elevated in presence of the cyclic AMP. So the cyclic AMP often releases or switches the protein kinase from a form of inactivation to a form of activation. And these kinases know how to phosphorylate, again with the use of the phosphate from ATP, and very typical residues. So we can actually divide them into two types of protein kinases: those that hydrolyze, that add a phosphate to tyrosine residues, and those that add a phosphate to serine and threonine residues on their target protein. Okay, so, and then the effect of the protein kinase A on the target protein is that, and also we talked about it a lot, that a lot of the functions in the cell, once you phosphorylate something, once you add like a phosphate for something, you actually charge it with energy, or you like turn it on. So it's like a switch effect, adding a phosphate. And this is exactly what these kinases do. And intracellular signaling in general can stop, or the termination of the cellular response can be either by lowering the levels of the cyclic AMP, like the secondary messenger, deactivating the protein kinase, or actually like removing elements of the receptor, or molecules that are related, or proteins that are related to the receptor from the plasma membrane, and thus terminating the response. Okay, so this is in very general terms of signal transduction. Now I'm not sure that you remember because it was very long ago, but we'll go over the questions from last chapter about transport of ion across the cell membranes. So before we continue, below are 3 steps from the operation mechanism of a protein that transfers an ion from one side of the plasma membrane to another. One is ion binding, conformational change, and release of ion in this order. So these 3 steps can be found among additional ones in the operational mechanism of what? So is it in a non-gated ion channel? What don't we find? I would say conformational change. Ion binding, you still have. We saw examples that you have an ion binding, but there's no conformational change. ATP-driven pumps. Yes. Symporters. Yes. Why not? Antiporters. And voltage-dependent ion channels. So the right answer is B, C, D. Okay, so in the voltage-dependent ion channels, you don't have like the conformational change is not, for example, it's not like in this order. That's what the conformational change is not dependent on the ion or something like that. So this is why it's not in this order and it's not included. But don't worry, I won't ask you questions that are like that tricky in the test. Is it clear? So, in what aspects do different ion channels differ between them? So the type of transferred ion. Yes. In the transfer rate. Yes. Dependency and independency on membrane potential. Yes. And dependency and independency of a ligand on ligand. Yes. So all answers are correct. During the transfer of a potassium ion across the non-gated potassium channel, or what we call the resting K channel, the ion is surrounded by water molecules. The ion sheds its water molecules. The ion interacts directly with other ions that pass in the channel. The ion interacts with other ions of the channel. Yes. So it's actually, we didn't show that it interacts with other ions of the channel. It interacts with oxygens of the channel, not with ions of the channel. So it's B, and I showed you the next one. Never mind. In the operational limit of the voltage-dependent sodium channel, the refractory period is achieved due to the protein global region that binds the channel gate. Which of the following is common mechanism for termination of synaptic signal? Slow diffusion of the neurotransmitter from the synaptic cleft. Yes or no? Is it common for the neurotransmitter to slowly diffuse from the synaptic cleft? Okay. Reuptake of the neurotransmitter? Phosphorylation of the neurotransmitter? Hydrolysis of the neurotransmitter? You shouldn't wonder about that, Nomi. Okay? So, yeah, it's B and D. So, most neurotransmitter are reuptake, but some are hydrolyzed, like acetylcholine. So, now that we're done with that, we'll start to actually talk about specifically on G proteins. And in this chapter, we'll focus on G proteins that affect the adenylytic class, which produces cyclic AMP. And then we'll focus specifically on G proteins that are related to ion channels and receptors, which are in this term unique to neurons. So, first of all, the structure of a G protein is what you can see here. So this is the receptor. This is a G protein coupled receptor, the ligand that binds to it, and the receptor is transmembranal. And the G protein itself is composed normally, but not exclusively, of three subunits: the alpha subunit, and the beta gamma subunit, which are often smaller than the alpha subunit. And the alpha subunit itself is what we call a GTP-as. Okay? And if we say GTP-as, it means that it breaks down or hydrolyzes GTP. And GTP is very similar to ATP, but just with a different base. So it's a high energy molecule, less common than ATP, but still it's an energy or phosphate giving molecule that the alpha subunit, we actually call it ATP-as, but it's a little bit misleading on the function of it. Okay, because the alpha subunit, it's not simply like an enzyme that breaks GTP to GDP. It's like a switch protein that has the ability to switch on, literally switch on other proteins that are close by or part of of the pathway. So the operational mechanism of G protein activation in general is, for example, if you have an inactive receptor, if it's not bound to any hormone or ligand, then the trimeric G protein itself is just floating around the membrane. It's anchored to the membrane because it has covalently linked lipids or lipid tails, so it's anchored to the cytosolic leaflet of the plasma membrane. And nothing happens in this stage. What happens when the hormone or ligand binds to the active receptor is that you get a conformational change. This conformational change facilitates the binding of the G protein to the receptor. And after the binding of the protein to the receptor, it opens the possibility or opens the ability of the GDP molecule that is bound here to be released and get exchanged by a GTP molecule. So when the GDP molecule is released and the GTP molecule binds to the G alpha, it causes a secondary conformational change that dissociates the G beta gamma subunit from the G alpha subunit. And now the G alpha is free to diffuse along the membrane. And again, I remind you that the membrane is something that we called like a fluid mosaic model. So it's there's almost free lateral diffusion along this line. And the G alpha is free to switch on or activate an effector protein. And a very common effector protein is the adenylytic class, but we'll give an example of that in a second. So, and then the G alpha the kinetics is very dependent on the pathway, but in general, the G alpha can activate many active effector proteins, but after some time has passed, eventually the GTP will be hydrolyzed to GDP, and the G alpha and G beta gamma subunit will reassociate once more. And if there is no hormone or and the receptor is not activated anymore, so this response will stop. And because it's complex, so I'll show you a quick movie that summarizes this.
Many G protein coupled receptors have a large extracellular ligand binding domain. Many G protein coupled receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound. In this case, a receptor kinase phosphorylates the cytosolic portions of the activated receptor. Once a receptor has been phosphorylated in this way, it binds with high affinity to an arrestin protein, which inactivates the receptor by preventing its interaction with G proteins. Arrestins also act as adapter proteins and recruit the phosphorylated receptors to clathrin-coated pits, from where the receptors are endocytosed, and afterwards they can either be degraded in lysosomes or activate new signaling pathways.
Okay, so this, in the end, they showed the end of the process of what happens when it's hyperactivated, but we're talking about it. We're going to elaborate about that in a second. So a small point is that G proteins are actually in 2 categories. We're only going to talk about the trimeric G proteins, like you saw before, which directly are activated by the receptors, but there is also a second type of monomeric G proteins, often called RAS, that have only one subunit of the trimeric G protein, normally the alpha subunit, and they're linked indirectly to the receptors through special types of adapter proteins. But in our examples, we're going to show trimeric G proteins. So in general, when we focus on the G alpha itself, molecularly, it has this switch-like operation. Like these 2 arms are actually pulled together by this phosphate group, that the last phosphate group from the GTP, and this is what we call an on state. Like when the G protein is in this state, it can fit into different clefts and regions in activator proteins and render them active. And when it's after the GTP is hydrolyzed, then the GDP remains, and in this state, in an off state. Okay, so it's really like a physical, you can like a spring mechanism. And there are a lot of different types of, like in the human genome, we have 27 types of G alpha proteins, 5 G beta and 13 G gamma subunits. Each one of them can activate a different effector protein, and in different systems. For example, the adrenergic system, the G alpha activates the RNA class that produces cyclic AMP. But in other responses, you can have, for example, in the visual system, then you can have activation of G alpha that will cause an opening of a potassium channel, and but we'll show specific examples. We'll show a specific example of these instances. It's just important to state that there are many types of G alphas, and each one of them has a different activity. And in some cases, there are rare cases in which the G alpha can also inhibit certain functions, and in more rare cases, the activation is done by the G beta gamma and not by the G alpha. So this unit is the activating element, and the G alpha is not. So when we talk about G protein coupled receptors, the most classical example is ones that affect an NAC class, like in the article of Kandel. And a more even classical example is the epinephrine example. So in this case, we're going to show what happens when the epinephrine responds as a model to show you how the body, if it's exposed to epinephrine, which is like a form of adrenaline, it can prepare your body to stress response. So first of all, when the body is stressed, it often releases an epinephrine that causes three major responses to this stress. You have increased catabolism of glucose and fatty acids that produce a lot of ATP, and this is modulated by beta-adrenergic receptors. So you have an increase in the levels of the ATP. You have increase in the heart rate contraction. This is again through beta-adrenergic receptors. And you have a cutting off of circulation of unneeded organs such as the stomach and kidney. This is part of the flight or fight response of the body, is mediated by these three elements, all of which are mediated by G proteins. So dependent on the type of receptor, a receptor that can detect or that will be exposed to epinephrine can cause an activation of an enzyme or cause an inhibition of an enzyme. Also, this typically doesn't happen in a specific type of cell. It will be in different types of organ. It will cause a different type of response. But in general, if you have a receptor that will cause an activation, you have a receptor that can cause activation of adenosine class and cause an increase in the production of cyclic AMP. So adenosine class is the enzyme that produces cyclic AMP. And on the other hand, you can have another receptor that will cause an inhibition of adenosine class by activating different types of G alpha. So this is a stimulatory G alpha subunit, and this is the inhibitory G alpha subunit that associates to this type of receptor, and it will cause a diminishing in the levels of the cyclic AMP by blocking this enzyme. So after the cyclic AMP is produced, it activates or it relays the signal that comes from epinephrine with protein kinase A that you all by now should know about. So binding of cyclic AMP to the R subunit of the protein kinase releases this subunit of the protein kinase, which has the catalytic site that can now go and phosphorylate target proteins. So actually, when we break down this response, we see that we have many levels of amplification of the signal. Okay, so a single epinephrine molecule can cause average activation of 3 different adenosine class enzymes, and they in turn, one adenosine class enzyme can cause a second level amplification, is producing many cyclic AMP molecules, and each cyclic AMP molecule can release one protein kinase A, and one protein kinase A can phosphorylate many target proteins or enzymes that will in turn produce many products. Okay, so we're not going past the activation of the PK in this example, because there's a lot of different, like I showed you, it depends on which cell in the body the epinephrine was detected by to know which type of cellular response happened. But what you can see here is that you have 4 different levels of amplification. Like a single epinephrine molecule causes a cascade of a very dramatic cellular response. But the downside of it, or maybe not the downside, is that all these steps take time. So it's a relatively slow response. And I don't know if I want to show the movie now. Yeah. So the termination of this response can happen. The termination of this process is caused by, or the modulation of this response, like I showed you, can either be that the receptor, the affinity of the receptor to the hormone can decrease after activation, and the cyclic AMP phosphodiesterase, which is the adenosine class, can be like removed from the cellular membrane or diminished from the cellular membrane. And if one of the methods that we see here, and what you saw in the video before, is that after prolonged activation, what happens is that we get a type of what we call homologous desensitization, which is that during the pathway of activation, and this is like the G protein coupled receptor that recepted the epinephrine, then you have a specific enzyme that was activated by protein kinase A. So you had the epinephrine before that caused the activation of protein kinase A. Protein kinase A, in turn, what it can do is that it can activate or phosphorylate an enzyme that's called BARK, and the BARK in turn is also a kinase, but this kinase can phosphorylate the receptor itself that the response started from. Okay, and this phosphorylation actually causes reduced affinity of G alpha or the G protein to the receptor itself, hence like diminishing the affinity, and causes what we call desensitization of the response. And when you have a prolonged response, an even longer response, you get an increased number of phosphorylations on the receptor, and also a diminishing. So it's kind of a way of the body to like take time and see how long the response has taken place. Because if the response is not prolonged, then the time by the time that the BRK is activated, it's already there is like the response is not very significant, and you need a long time of activation in order for the BRK to be active and start phosphorylating. And if you have even more phosphorylation, like in extreme cases of prolonged activation, what will happen is that beta-arrestin that can bind to this phosphorylation site or associate to this phosphorylation site completely like puts the last nail in the coffin of this activation, and can cause, first of all, like there is no way that the G protein can associate to the receptor now, and also in some cases it will cause endocytosis. So the entire membrane, together with the receptor, will be incorporated inside the cell, and even the receptor itself will be degraded. So it's kind of a feedback mechanism of the body in order to regulate these responses, and not for them not to be too and like retain a level of sensitivity of the system, but not too much sensitivity. So where is it? I hope that this is this movie, but I also found a nice movie that summarizes this entire response.
Imagine you are hiking through the woods on a late summer's eve when you Imagine you are hiking through the woods on a late summer's eve when you have the misfortune of coming across a rather disgruntled grizzly bear. Given the scarcity of food in these parts, he is delighted to have come across such a palatable entree. A series of transient depolarizations in your neural circuitry identify the bear as a predator and quickly decide that running away is your best option. In order to elude your pursuer, you'll have to react quickly at both the synaptic and cellular level. To accomplish this, a number of signaling molecules are released to various targets throughout the body. When faced with this type of stressor, the brain sends a series of messages that lead to activation of neurons in the peripheral nervous system. This leads to release of a neurotransmitter called norepinephrine. But how does this chemical signal get translated to a cellular response that will allow you to escape? The effect of the chemical signal depends largely on the type of receptor to which it binds. Ionotropic receptors are located in the cell membrane and have a pore that allows ions to pass through. Once inside the cell, the ions can alter the local membrane potential and increase or decrease the likelihood of an action potential. A different class of receptors is the G-protein coupled receptor, or GPCR, which are part of a group known as metabotropic receptors. As the name implies, these are paired with a G protein, or guanine nucleotide binding protein, which is composed of 3 functional units, an alpha subunit and a beta-gamma dimer. Normally, the G protein is bound with a guanosine diphosphate, GDP molecule, and is inactive. Unlike ionotropic receptors, the GPCRs have no pore. Rather, their efforts are mediated by activation or inhibition of other molecules. When a signaling molecule, for example a neurotransmitter, stimulates the GPCR, the receptor essentially changes its shape, allowing the G protein to bind to its intracellular domain. The alpha subunit then exchanges its GDP for a guanosine triphosphate, GTP, creating an active state. This results in the G protein subunits dissociating from the receptor, as well as each other. The alpha subunit then binds to an effector protein, such as an enzyme, which then acts to increase or decrease the production of a second messenger. These so-called second messenger pathways can lead to a number of possible changes inside the cell. This is perhaps the main difference between ionotropic and metabotropic receptors. While the effects of the GPCR are typically slower than those of the ionotropic receptors, they are much longer lasting. An astounding number of second messenger pathways are found in our cells. One example is the cyclic AMP-dependent pathway, which, as in our example with our hungry bear, may be activated by the neurotransmitter norepinephrine. In this case, norepinephrine binds to a specific kind of GPCR called a beta-adrenergic receptor. Once this receptor is stimulated and the G protein activated, the alpha subunit binds to an enzyme known as adenylyl cyclase. This in turn increases the production of cyclic AMP, which is the second messenger. Cyclic AMP will go on to activate protein kinase A, pKa, which in turn regulates a number of different proteins. Ultimately, activation of the cyclic AMP pathway may influence processes such as cell survival, the up or down regulation of gene transcription, and the production of several neurotransmitters and receptors. Now, back to the bear. At the moment that you first encountered it, your flight or fight instincts decided that it was in your best interest to run. The activation of diverse pathways in the brain ultimately results in a surge of norepinephrine, which binds to the beta-adrenergic receptor located in many of your organs. This causes your pupils to dilate, your heart rate goes up, and blood circulation is increased to your muscles to push them to their limits. It does this through activation of intracellular signaling pathways, such as the cyclic AMP pathway we have just seen. So, by binding to this specific GPCR, norepinephrine allows you to escape, despite the odds.
Okay, so luckily now you'll understand what you need to do, what your body does when you encounter a bear, but it happens in many other times. And the last example for today of G proteins, and the first example is also the previous example was in neurons, but this is more a classical neuronal example, is the way that G protein coupled receptors can regulate the function of ion channels. For example, in the acetylcholine example, we have what we call we talked about the nicotinic receptor, which is an ionotropic receptor that just opens a channel once it's bound to acetylcholine and allows permeability of sodium and potassium. In this case, the binding of acetylcholine to a receptor that's called the muscarinic acetylcholine receptor does not cause the direct opening of any channel, but activates a G protein. That this G protein can now associate. In this case, this is one of these rare examples where the G beta gamma subunit is the one that does the work. So it associates to a special type of channel that now opens and allows permeability of potassium. And this is, for example, what happens in the heart muscles that causes once acetylcholine is being recepted by it's through the vagus nerve by the heart, that it will cause a slowing down of the level, slowing down of the heart rate. Okay, so for example, you can think about this response as some kind of response that is more slow and more modulatory. Okay, it's not like something that is very dramatic and very transient. It's a slow and modulatory response that just helps induces a slight causes hyperpolarization of the cells, and thus causing the muscles or the heart muscles to slow down their activity. Yeah. Yeah. It's yeah. Yeah. G proteins, they're found everywhere. Okay, so in the parasympathetic and the sympathetic. The previous example was epinephrine, which is normally sympathetic, and acetylcholine is something that you can find in the parasympathetic, but also in the sympathetic. It depends. It really depends on the type of receptor that the cell has. Like if the cells would have like a ionotropic receptor, then it would be completely different, and or a different muscarinic receptor, then the response can be gene transcription, for example. It doesn't have to be sodium channels, potassium channels opening. And I told you that GPCRs, one of the things that they do very well is amplify the signal. So in the case of epinephrine, you saw that we got a very dramatic response from a few epinephrine molecules. In another system where biology chose to induce amplification is the visual system. So the rhodopsin in your eyes, you probably heard that name, but you didn't know that it's a G protein kappa receptor that is activated by light. So it's localized in these. This is an EM image of a rod cell, a human rod cell that is sensitive to low light conditions, and it is responsible for black and white vision. And inside these discs that are inside the cell, you can find a high concentration of these rhodopsin proteins. And the thing that is interesting about this system is that if before we talked about a physical, like a molecule ligand, in this case we can think about this G protein kappa receptor as if the ligand is already bound inside this receptor, and light, what it does is that it changes the conformation of, in this case, 11-cis-retinal, which is a derivative of vitamin A, from a cis conformation around this 11-12 carbons to a trans conformation, and thus it causes so that the light or a single photon that hits this retinal molecule causes a conformational change that triggers the conformational change in the entire receptor. So the ligand in this case is already bound, and the light is what causes the ligand to be detected or to cause a conformational change in the rhodopsin. And as you all know, this molecule is very important for it's a derivative of vitamin A, and vitamin A is a derivative of beta carotene. So this is why it's very important to eat a lot of carrots and this is why you all know that you never saw a rabbit with glasses, right? So you know that. But actually, I can tell you that in the past few years I looked online and I found many examples of rabbits with glasses. So I don't think it's very, I don't know, maybe it didn't eat so much carrots like it should. But, so, and this is like the model that we know, this entire structure, like the association of the G-alpha and the rhodopsin itself, wasn't completely deciphered. Like we don't have a very good crystal structure. We just have the crystal structure of the rhodopsin itself, so a lot of it is estimation, but we assume that this is like the type of association between the G protein. Again, this is the GTP and the G-alpha and the G-beta gamma, and these are the sites that are anchored, that have lipids that are anchored to the plasma membrane, and this is how they associate with the rhodopsin receptor. So how exactly does this mechanism happen? During the dark, the rod cells are in resting potential of -30 due to the fact, and this is different from other cells in our body, which are normally -60 or -70, due to open cyclic GMP activated non-selective cation channels. So the cell itself is filled with a lot of these channels that are constantly open when they're associated to cyclic GMP, which is in high concentration during the dark. So the cell is always left in a very depolarizatory state, meaning that it releases, if you did a little neuron science by now, that it constantly releases neurotransmitter. Okay, so the rod cell is like a cell that, in contrast to other cells, it constantly releases packages of neurotransmitter. But once a photon is recepted by the opsin, what happens is that you get a conformational change in the opsin. The G protein becomes activated, like I showed you before, travels along the membrane and activates an enzyme that is cyclic GMP phosphodiester AS. Okay, and the AS tells you that it breaks down cyclic GMP to GMP, this enzyme, and what it does, it lowers the local concentration of cytosolic cyclic GMP, and thus closing these channels. And closing of this channel causes hyperpolarization and the decrease of the release of neurotransmitter. And the actual reduction of the neurotransmitter release is what is the signal in the case of the visual system. And it looks very inefficient, right? This whole process looks like very strange, and very slow. And it's actually, in molecular terms, it's very slow. Like they say, it's said that this entire process takes can take up to 20 milliseconds or 10 milliseconds to happen. And this is why also our vision is something very slow. You know that you can't detect something that is above 50 hertz. So 50 hertz is like 20 millisecond difference. And this is about the differences or the possibility of our system to detect light. But what it does do is that it allows a signal photon to create a very large cascade of a signal, and this is actually what helps our visual system to detect the signal photon. And in next class we will elaborate about these the orders of magnitude. But in general, you should know that human vision is something very extraordinary, especially in the possibilities of our visual system to detect different intensities of light. For example, we can see very good in full sunbeam, but we can also detect single photons under very partial moonlight. So, and the way that this adaptation occurs, that is like 9 orders of magnitude as a physical signal, we will talk about next class, and we'll show examples of how exactly rod cells adapt to different levels of ambient light. But I'll just give you a hint that it has a lot to do with the modulation of the different components of this system. Okay, so see you next class, and we'll finish this chapter, and then